those of you who know me, I go sort of through these cycles every 5 to 6 years where I ask myself, do I do the right thing? And what could I do different? And so I just went again through one of these cycles and decided to move back to Switzerland, where I grew up and escaped 25 years ago, so 5 cycles back, because I didn't like Switzerland, and now I'm pleased how it is actually in Basel. So give me a call when you come to Basel. I also switched now to the other side from academia into industry, and it's really fascinating. So I'm actually back in this moment in my life where I learn every day something really new I did not expect. Okay, what I'm going to talk about today, though, is not from Basel, obviously. I just moved 3 months ago. We're still unpacking 165 boxes. But it goes back to 2 cycles in my sort of crisis evaluation when I moved to Texas, right? And so all the work I'm going to present today was done at UT Southwestern Medical Center. And I got permission from Alex to actually not show much math and modeling, but to sort of tell you what you guys could model when you get bored in your own futures and you have your own evaluation cycles. And what I'm going to stimulate here is really the idea. We have talked a lot about cell shape. Konmu gave a fantastic talk how important and predictive cell shape is. Cytoskeleton organization, what I'm going to try to illustrate today is actually this is not only downstream of all these signals that regulate these mechanical processes, but that the cell shape and cytoskeleton is probably directly involved in the regulation of signals. Okay, so here was my crisis 12 years ago. I spent, as in the introduction said, writing trackers and all these kinds of things on cells that look like that, right? Flatland cells, and we saw many of those already in the meeting, right? And what you see here in this little cartoon is 3 images of the cell type. It's not exactly the same cell, but it's the same cell type. It's a melanoma, where you have the morphology on glass, you have the morphology when you put the cell on a collagen gel, which still the cell feels the glass, and here's a collagen gel that is too thick for the cell to still feel the hard surface. And you immediately see that the morphologies are completely different. Right? So I realized that I studied the wrong thing for 15 years of my academic career and said, okay, I need to change this. And so Texas gave me this little pile of money to actually recruit real smart people. Reto Fjolk and Kevin Dean, who built microscopes. That was really the beginning of the light sheets, and they built these incredible light sheet microscopes which have isotropic resolution, X, Y, Z. Eric Welf came as the experimentalist of the team, and then Megan Driscoll, many of you might know her, was the first sort of physicist, computer scientist who took on now this 3-dimensional data. And what you see here is the raw data of a dendritic cell sitting deep inside a collagen gel. And on the right side, you see the analysis and the color code. I'm not asking you that. Typically, I ask math audiences, tell me what this is. It's actually the curvature encoded. The reason I bring this to your attention, the resolution of these microscopes is so incredibly good that for the first time, I think in sort of the literature, we were able to do really differential geometry on the surface of a cell. So we could really quantify with incredible precision these curvatures, and this is all Reto and Kevin who deserve that credit. Right? Okay, you can do that not only for a single cell. So here I don't show the collagen anymore. So this is a lung cancer spheroid growing again away from any mechanical perturbation. And it's about 150 cells here. And this is an isogenic model. So we know that the genomic profile of every single cell is identical, but you see a broad morphological heterogeneity. And when you now go with a standard of care drug and treat this spheroid, then depending on the morphology, you survive or you die. And of course, this is the bad news for our clinical oncologists, right? That's something they actually do not really think about, right? And so form follows function, right? So my next crisis is going to be in 10 years when I want to retire, and I already have that Corbusier chair, some Swiss, I love Swiss architecture, right? So that's where I'm going to sit because the form is unbelievable. It exactly fulfills the function for me sitting, right? Anyway, we know form follows function very well from Darwin's finches. I don't need to elaborate that. You all are familiar with that from high school. And more interesting, and I think it was alluded to already in this meeting, and more than 90% of all cancers, despite genomics and despite all this fancy stuff out there, are still diagnosed at this level, right? You have a pathologist who gives you an average 10 seconds. Be aware of that. You get 10 seconds. They make cells are clear with apical and linear orientation of nucleus. And then they say, okay, what happens to you, right? And here's just examples for as the cancers get more miserable, right? So I don't need to belabor that. Okay, so it's clear in the clinic morphology matters. However, when we now begin to think about that experiment I talked about, what is the relationship between form and cancer cell function, that's not so clear. There's a lot of examples. Bakal, Wirtz, Lee, et al., long list of people who have unleashed machine learning to show that form is predictive for the core functions of a cancer cell, namely proliferation, survival, and maybe migration, but the relationship is largely unknown. And that's what I would like to discuss with you. Right? So the inspiration for some of these studies I'm going to describe really came again from movies like that. This is now a melanoma cell, again sitting deep inside the tissue. And what you see here color-coded is now not the concentration of a kinase, but the activity of that PI3 kinase. And for those of you who are familiar with cell signaling, PI3 kinase is one of the key drivers of cell survival, right? And so when we watch now this movie, then you see here, there's a bubble coming out, and then the cell gets red. And I'm not saying that time is causation. I'm not going to go there, right? But it still triggers, hopefully, you're thinking a little bit that maybe morphology might actually control that upregulation of that PI3 kinase signaling locally there. Right? Okay, back to Megan. This is sort of our high school Wednesday afternoon exercise to really scared biologists. Right? What we have here is a simple cell with two molecules illustrated and the third one not illustrated, which has receptor tyrosine kinases sitting in the membrane, and they activate blue molecules into greens, and then you have a deactivator in red. Right? And so you can have these stochastic simulations, and you see this is a perfect, nice mixture of a globular cell in 3D. The big problem here is when you culture this cell on glass, which is what many of you have shown us in this meeting, and analyzed to death. The problem is that just by the sheer fact that you culture this cell on glass, you upregulate the activity of the signal by fourfold, right? And so you stare at me. I don't know, are you a biologist? It's nerve-wracking, right? That's a problem, right? So I have audiences screaming at me. Okay, and then the next problem is you see actually now the signaling activity gets subcellularly spread, right? So the flatter you are, the more signaling you have, right? Okay, well, it's a completely trivial law, right? Many have pointed that out, but I think the experiments to really follow through with that are only possible now today that we have this 3D microscopy, right? So what we changed here from going from globular to flat is simply the surface to volume ratio, and so the biochemical landscape absolutely changes, right? And so now you can add all these morphological motifs. Von Mu segmented beautifully, right? Here are his little philopodia, which he can't segment. That's why I asked you about that, Von Mu, right? But all these morphological properties of these cells act as diffusion traps, as signaling activators, as scaffold assemblers. And we also talked about mechanics, right? So if you push on something to protrude, well, you actually get the mechanical input that again triggers a signal in many different ways. One way described beautifully by Margaret Cardell, right? So you have integrin-mediated adhesions. Okay, so all this comes together as this morphological control soup that is in a highly nonlinear interaction with signal transduction in these cells, right? Okay, and so the proposal I'm going to make and then show you hopefully now some evidence is that we study signaling going into cytoskeleton architecture driving morphology. And we all know, and I'm going to come back to that throughout the presentation, the environment feeds into the signaling. The environment through dimensionality and mechanics, stiffness, for example, controls how the cytoskeleton architecture translates into morphology. Right? What I'm proposing is that morphology has a direct feedback into the signaling, and that in the case of cancer, I'm very interested in cancer, in the case of cancer, this change coming from the morphology in signaling is a key factor in proliferation and survival of cancer cells. Okay, this is of course a huge causality issues, and I could give a whole talk about causal inference. This is something I've been working on for 20 years. Series of papers here I want to point out. Von Mouly in 2015 brought us one step closer. So we're moving essentially out of academia into Wall Street, because Wall Street has exactly that problem. You cannot perturb your system to get the causal flows. And that is the same thing in this system here, right? It's very, very hard to perturb morphology in a causative way to test whether you actually influence signals. So we need these sort of financial math tools, right? And so we're getting there, right? So if you follow our papers, if you like to hear more about that, it's getting pretty cool what one can do with these tools. Okay, not going to talk about that. Let me go into a story which has been published about 4 years ago, but it's sort of a nice segue for the rest of the presentation. Asaf Zaritsky, he has been mentioned on a few slides also today. Computer scientist now in Israel, Andrew Chambers, and also computer science, and Eric Welf worked together on this question: is there any evidence for shape control over metastatic cancer cell function? And it is, again, we have heard a variant of that already before today, a story of GenAI. Okay, the system we worked with is Sean Morrison at UT Southwestern has this incredible system where he has melanoma tumors passaged only in mice. So absolutely you can never see a flat plastic surface, right? Okay, and we learned how to take these tumors out of the mice and disaggregate them. So we have single cell movies of the melanoma tumors. And Sean has shown in this paper that he has more than 90% correlation between what happens in the mouse 10 years after the tumor has been removed from the patient and what happened to the patient. So we had a pretty good ground truth to train this system. And so one of these two tumors, I don't know which one is which, is highly metastatic, and the other one, it's a tumor, so it grows, but it is less metastatic, right? And under these phase contrast microscopes, you can see these cells wiggle, and von Mu could now analyze the heterogeneity and maybe find the signal in there, and the signal is actually spectacular, yes. So you can, at the tumor level, 100% accuracy, distinguish high from low propensity metastatic tumors. It doesn't work at the single cell level. Von Mu told us that already. There's too much heterogeneity in there, so that the distributions overlap a little bit, right? Okay, how is this done? This was done with a variational autoencoder where we extracted, in this case, a 56-dimensional auto latent space. And then we used that latent space as the dumbest possible classifier to now distinguish between high and low metastatic cells. The autoencoder was trained by about 1.5 or 1.7 million images, not only melanoma cells, but other cell types, right? So it's a completely generic latent space description. Okay, good. So I wanted to validate that thing. So we had from each PDX, so Sean's tumors, we had a classifier, they're all highly consistent. And now we ordered from ATCC melanoma cell lines, but they don't know anymore that they're cancers, right? They have been abused by human cell culture for 30 years. Right? Nonetheless, very systematically, we find one cell line, A375, which is a very famous melanoma cell line, as highly metastatic and MV3s as low metastatic. So we stuck this into the mice and then took the organs out and looked with bioluminescence, and sure enough, A375s are highly metastatic and MV3s are not metastatic. So PDX trained on cell line into the mouse validated, pretty good. And what is even more amazing is the primary tumor grows for both cell lines exactly the same way. Or if we look very carefully, it is actually the less metastatic tumor which grows even a bit faster, right? So obviously what this latent space encodes is a very specific piece of information about metastasis, which is not the same as tumor growth. And everyone in the cancer field knows that, right? But what is magic here is that the AI sorted this out, right? And we haven't really talked too much about genomics, so I want to empower people who think about cytoskeleton and morphology. This is your empowering slide, right? We took now and sequenced all these cells, and there's absolutely no signature on metastasis in the genome. I'm not going to go through these graphs, just believe me. You can read the paper. Okay, so what's fascinating is that this autoencoder captures a metastasis-specific image property. What is it learning? And here comes now the power of Gen AI. And I don't want Erin did a fantastic job in explaining all that. The only thing I want to add here is that with Gen AI, you can actually go and hallucinate. And the flip side of hallucination is discovery. So what we do here is we walk now in that latent space from low to high metastasis and a little further and generate movies that show us maybe what these cells do. And surprise, surprise, cells with uropodal extensions are more metastatic than those with less, and you can turn now an efficient metastasizer into a non-metastatic cell. Of course, the migration people immediately say it's trivial. This is migration. It's not migration. It's absolutely not migration. And we have seen that theme several times already, right? I think Kwon-Mu also showed an example. It's the elongated cells which are more aggressive, right? So we find here in a completely unbiased manner this behavior, right? Okay. We actually continued that work now. If you're interested, this is on bioRxiv. Here, in this case, we observed what the Gen AI delivered and then interpreted. We have now these so-called delta marches where you go in the latent space and do very, very infinitesimally small steps. And then you can actually automatically extract what the rules are. So I think this is a next cool step. Okay, so the prediction here is uropodal extensions drive specifically, again, remember, the primary tumors grow equally fast, right? It's a specific signature of a pro-metastatic cellular program. So what is that? Well, in parallel to this AI story, Abby Mohan, who was an MD PhD student, discovered these killer lamalipodia in melanoma. So this is a melanoma cell line which expresses a RAC mutation, contrary to RAS, where we have constitutively active mutations in all cancers, or in many, many cancers, 50% of cancers. These RAC mutations are not constitutively active, but they're hyperactivatable, right? So all you get is a little stronger RAC signal. And they make these massive, massive, massive lamalipodia. So what's going on here? I'm actually going to jump over this. What's happening in the clinic is that patients with RAF transformed melanoma who express that mutation, it's about 7%, they're completely insensitive to any therapeutic intervention. Okay, and so we wanted to study that at the cell biological level, right? And so the interesting thing here is, here's a RAC wild type with no drug, so this is the typical wiggling. The projector is a little strange here. And when we go now with MAP kinase inhibitor, which is standard of care chemotherapy against these types of melanoma, you already see there is a little bit of a lamalipodia formation. But when they now have P29S, that mutation of RAC, then you see that you extend these massive lamella-podia. And I'm summarizing now a long, long story, which is all published. What happens is that you extend lamella-podia beyond the normal extension of a lamellipodium. And in there, you isolate now PAC, which is a kinase directly downstream of RAC, and NF2, Merlin, which is a very classic tumor suppressor, which has an actin binding site. And because these lamella-podia are chock full of actin-branched actin, and so narrow, you have essentially a vacuum cleaner that pulls the tumor suppressor away from the nuclear areas where they should act as a tumor suppressor. And in addition, PAC actually phosphorylates Merlin, and in this case, phosphorylation is inactivation. So you have a double whammer going on, completely morphologically driven. By making these lamella-podia, these cells essentially take the tumor suppressor function away and therefore can proliferate despite the drug challenge. Okay, very interesting. Linking back to the AI story. Again, primary tumor growth with or without P29S mutation in RAC is identically fast, but you see again with P29S, you have a highly metastatic situation. So it's not only protecting against the stresses of chemotherapy, but protecting against the stresses of metastatic travel. And we showed again then in histological sections that it is really the proliferation, not the migration, it's the proliferation which gets upregulated in the metastatic colony. So it's a pro-proliferation program, these morphological features start. Byron Weiss, in a publication that just came out this year, wanted to visualize these signaling zones a bit better with biosensors, that's that. And then he thought, okay, I can have just knock out NF2, the tumor suppressor. And to our surprise, what he found is that when you knock out the tumor suppressor, again, you make massive lamella-podia. And when we go and put now in the knockout cells Merlin back, so re-express the tumor suppressor protein, then all of a sudden that signaling zone disappears, which means that not only do we have that double whammer from RAC, PAC, Merlin spatial dislocation of the tumor suppressor, but the tumor suppressor itself is potentially an inhibitor of that morphological program that drives its inactivation. Mother Nature is unbelievable, right? Okay, so that's what we found in this data. And so this is sort of the summary of all that study. This tumor suppressor really triggers a double negative feedback, and all the signal analysis people in the room know double negative feedback is the most stable scenario of actually activating something. And this tumor suppressor completely controls its morphology and the downstream signaling in these morphologies. And of course, from a cancer perspective, again, the question is, are actually tumor suppressor really transcriptionally regulating tumor suppression, or are they controlling morphological programs which lead to tumor suppression? That's a question. I have no answer yet. Right? Okay, I want to come back to that a little later with another story, but before that we go into a second little side story where we put epithelial cells again on stiff and soft substrates, and everyone in the room knows on stiff substrates cells make stress fibers. In soft substrates they ball up and have more diffuse cytoskeleton arrangements. What we and probably you didn't know before this study was that with this you change actually the metabolism of cells, right? So cells on soft substrate have much less glycolysis. Not so cancer cells. Cancer cells don't care about soft or stiff. So they pump up their glycolysis, and of course, you might all think of the Warburg effect, right? That's what's going on here potentially, right? Cancer cells maintain their glycolysis, right? Well, what's the difference between a cancer cell and the non-cancer cells? So here are normal lung cells, lung epithelial cells, and here are lung cancer cells. Well, cancer cells maintain stress fibers and very thick cortical bundles also on soft substrates. And this is something that has been described before us. It just took a long, long aha moment for us to realize what's going on, right? So what's going on, it's a paper published in 2020, is again the cytoskeleton completely controls the rate-limiting steps of glycolysis. By having a phosphofructokinase, which drives really the glycolytic pathway. So when you have stress fibers, then you have this TRIM21, which is an E3 ligase that would tag phosphofructokinase specifically for degradation. The cytoskeleton, again as a vacuum cleaner, pulls essentially the degradation molecule, the degradation regulator away. And when you now go onto soft substrates, the stress fibers disassemble, and this E3 ligase gets unleashed, diffuses around, and now shuts down glycolysis by tagging the kinase for degradation. And of course, in cancer cells, these stress fibers never go away, and therefore you have high glycolysis. And that's exactly what a cancer cell, a tumor cell needs to efficiently grow. So cytoskeleton dictates the world here, right? Okay, I hope you begin to believe me. This is an interesting problem. So the last thing is a paper we published in 2023, which is about blebs. Again, here, surface curvature encoded in color. And now we want to get a little more quantitative. Actually, amazingly, these curvatures are about 1 micron radius, right? 1 micron is the magic length scale in biology. I ask that all my students, right? Why is a bacterium 1 micron and why is a mammalian cell 20 microns? Bacteria do not want to deal with diffusion control, but mammalian cells do. Right. Okay, so a melanoma cell bubbles, technically called blebs, in these random little protrusions, and then they get pulled in. Very well described biophysically. These blebs form when you have little cracks in the cortex or in the cortex membrane attachment. And since the pressure inside the cell is higher, you blow a little balloon, and then actin polymerizes, and the myosin comes in and pulls it back, right? And that happens on a time scale of about a minute or two, right? And these blebs have been studied ad nauseum in the context of migration, right? It's the driver of proteolysis-free pathfinding, like shown in this beautiful movie by Eric Sahay. It's really got popular as a low adhesion migration mechanism, and it is clinically known to be very much associated with aggressive metastatic spreading, right? Here's my last migration paper. I thought I should show that quickly. We actually found that these blebs are also not only for pathfinding, but good for path generation. They actually box very steadily. They box the material away. And to have that persistence, we had to sort out the positive feedback between the bleb and signaling. I'm not going to talk about that, but it's yet another example where morphology completely controls the signal you need to make a new bleb, right? Okay, what is interesting now is you take a garden variety of non-cancer cells and you trypsinize them and they lift off and every cell starts to bleb. And these are not apoptotic blebs. These are just dynamic blebs, right? Zoomed in here nicely to show you that, right? Okay, we all know that cells to be happy need either friends or a substrate. And that is because Dr. Gaudenz studies integrin-mediated adhesions, which give that signal that they need to survive and proliferate, right? Or others study cadherins, which do the same thing, right? And when you now actually trypsinize these cells, then they lose that input and undergo a form of cell death known as anoikis. And probably everyone who had ever a biology course knows that a hallmark of cancer is that cancer cells can survive in this state. hopefully the bells are already ringing, right? So cells begin to bleb when they are in this state. Well, maybe these blebs are a defense mechanism for cancer to survive, right? And that's exactly what I'm showing you with this thing. A normal fibroblast blebs for about an hour or two, and then endocytosis is a little stronger than exocytosis in these cells. The membrane gets too tense, and the pressure does not allow these cells anymore to blow out blebs. While a melanoma cell, and I have no answer why they can do that indefinitely, but they do that for a long, long, long time. They maintain their blebs. So a fibroblast in suspension dies after about 2 hours. A melanoma cell and many, many other cancer cells are anoikis resistant. Okay, so the question is, of course, are dynamic blebs survival organelles? And now I come back to that movie I already showed you. PI3 kinase is a survival signal, and the bleb seems to control that PI3 kinase. Okay, so we studied that in more systematic ways. We had, through Megan Driscoll, a beautiful computer vision tool to quantify blebs. So you can, in this case, we actually are lucky, we can manipulate the morphology a little bit by actually gluing the cells up with germ agglutinin. So we go at relatively low concentrations and take the blebs away. And then you see now here MV3 cells, when they are adhered and we increase the good glue, they don't really care. But when they're detached and you go now and take the blebs away, they begin to die. And I'm not going to belabor that here too much. This will be a super interesting side story. It works. It just works. Okay. Okay, so how would dynamic blebs promote survival? And this is now one of these universal themes. I just showed it for now with detachment, but you can stress cells in many, many other ways, and they always begin to bleb. Okay, and what these blebs do, remember what the curvature was? 1 micron, right? 1 micron. Well, there is a protein, one polymer family, that is sensitive to negative curvature of 1 micron length scale, and that's septins. And we know that from yeast, right? At the yeast bud, we have septin rings. And what we now have here is in mammalian cells, these septin rings. That projector, mathematicians really need to buy better projectors, right? So that biologists can show their data. Okay, they have the cartoon. The septins sit beautifully exactly at the bleb necks, right? And here's a three-dimensional visualization of that. And now what these septins do is they recruit the oncogenic RAS. So the mutated RAS, keep that thought, the mutated RAS to a signaling scaffold, which now efficiently drives survival signaling through MAP kinase signaling et al. Okay, what's so astounding here is that I tell you these cells contain a mutation which would be clinically identified. But when we take the bleb away, we're not taking the mutation away, and we're not having lower expression of that RAS oncogene. It is the spatial localization of the oncogene inside the cell which makes oncogenic penetrance. And that is a very inconvenient message to our colleagues in the cancer world. That's something they really have not yet appreciated. Okay, I hope you're excited because there are now real modeling questions, and that's what I wanted to show you, right? The hypothesis here is, of course, blebs promote curvature-sensitive scaffolds. That we have already experimentally shown, and we can also sort of, in a crappy Wednesday afternoon model, show that, right? But what is now unbelievably interesting, and think again, you cannot perturb blebs. So that needs modeling. This is now not a model which will sort of restate the things the experimentalists have already seen. This is going to be a model which is really predicting a new mechanism, potentially. You make a scaffold next to a bubble, which is potentially also a diffusion trap. And so what we need is a model that goes now into the nonlinearity of RAS, PI3 kinase, and all these feedbacks that come, and show that it is the bleb formation not only to trigger the signal, but to amplify it. And I'm not going to do that. I have changed my career. So go do it. I would be really thrilled, because that might actually really change the way we in pharmaceutical companies think about cancer. Okay, so I told you about the fibroblast, which undergoes anoikis. Well, as soon as the fibroblast can no longer make blebs, the septin is diffuse. Again, this is not an expression. The septin concentration is absolutely identical in these cells. It's a matter of, do we polymerize or do we not polymerize, right? And septin needs negative curvature to polymerize, and the round cell doesn't offer negative curvature. But when you look at the same fibroblast 30 minutes after detachment, when it still can bleb, then it makes the same septin scaffolds as the cancer cells make. Okay, here comes the moonshot experiment. We were wondering, can we take a normal, stupid fibroblast who has no clue of cancer, who has normal wild-type RAS, non-mutation, and can we push that fibroblast to become anoikis-resistant, so acquiring a hallmark function of cancer, by just prolonging blebbing? Okay. And the answer again is yes. I'm not going to go through the data. Yes. So you can turn a non-cancer cell into, I'm not saying that these cells make tumors. I want to be very clear. But you can turn a normal cell with no oncogenic mutation into believing it is a cancer cell for a little while by changing morphological properties. Okay, so that was published 2 years ago, right? So, of course, and that's exactly why I did now switch side. These are beautiful cell biological studies, all in 3D. At least we're 3D, right? But the question is this all just an artificial behavior of bottom-up cell biology, right? And so the way to go after that is you need to build real tissues like organoids. And many, many academic labs build organoids. But the big hard thing is they're very hard to make them reproducible so that you can do large-scale experiments, right? And that's exactly what the IHB has managed, right? So we have an organoid farm, and we can grow 1,000 tissues a day to actually do these types of experiments now and go after that, right? So Andrew Weems, who was the first author on that Nature paper, has done some experiments in this direction already in mice, not in organoids. It's all the problem of mouse experiments. And what I told you as single-cell biology is we see it in the mouse. So I'm not going to show the data. Stay tuned. The paper should come out next year. Okay, so here is my summary. You saw that. Now it's no longer a dashed arrow, but it's a solid arrow. I hope I have given you 3 clear stories where this happens, right? And a 4th story, you can read up on it, bioRxiv. Margaret might be very interested in that. Facin or actin bundlers in general actually assemble things which look like adhesions but have no integrins. And they, again, allow now cells to proliferate in low adhesive environments. And facin is known as an oncogenic driver, right? And then a big Ming, I couldn't agree more with you, guys start to work on intermediate filaments, right?
Intermediate filaments.
Right, because in this paper by Ben Naines, published, and he has a lot of follow-up stories, is what we found is keratin has 56 isoforms. Why does a knucklehead polymer like an intermediate filament need 56 different isoforms genetically, right? Well, the reason is, depending on which isoform you stick into these keratins, you begin to accumulate different kinases, which completely change the signaling landscape. That, colleagues, is interesting to model. Okay, let me conclude with this to again empower you. We are, as sort of biophysicists, always pushed into the corner by these guys, right? So I'm going to push back. And Kwon Mu, I was very disappointed that you switched the field, right? Go back to your images. Forget this scRNA, right? So genome goes into transcriptome, goes into proteome, signaling state, metabolic state. The bad news for all the transcription measures is that here comes the environment in, right? For the first time. How the proteome gets translated into signaling states and metabolic states is environmentally sensitive. You have a little bit of noise everywhere also. That's the headache of these guys, right? And then you have downstream of that cell function, right? The genomics people just draw that arrow here, right? And all these pathway studies from here to here, I cannot see it, right? And then we have morphology, and I think we have now established the way how morphology and function works. And of course, the beauty again is environment directly influences how the signaling state goes in the morphology. So from a control theoretical point of view, where do you want to sense as a cell what's going on around you? Certainly not up here. Certainly not up here, right? Okay, here we are. That's the magic bullet. And with that magic bullet, you control actually all the things the single cell sequencers see downstream of morphology. So that's what I wanted to show you. Any questions?
Yeah, this is your final moment of deviant.
My last statement right now. Okay, please.
Really nice talk, and I really thinking about that, the biophysics of to go back to your findings of living enabled survival, are you, since it's a non-additive environment, are you thinking in the context of circulating tumor cells?
Yeah.
Where the tumor cells have.
Yeah, so that's exactly about all the mouse studies now. It is amazing, right? So we can manipulate septins in the mouse. And what you blow away is micrometastases. And probably, so we haven't shown directly circulating. But when you need to survive as a metastatic cell, you have only one sorrow. And that is, how do I survive with no partners? And that's exactly, we have IP on that, right? So that's potentially a magic bullet really against metastatic disease.
Right? Follow-up question would be then, there's been a lot of work showing that the clusters are much more metastatic compared to
right.
single circulating tumor cells. I wonder how do you see clusters?
Yeah, I mean, I think that's completely, I completely believe these anti-eval stories, right? So you can either say, I bleb and have this survival, or I have still some friends as clusters. It's the same theme, right? You just need that signal. And how you get there, cancer cells will figure it out, right? Other question? Yes.
You know, you opened your talk with the pathologist simplistic view. And I want to know,
I did never say simplistic view.
easy assignment of
Yeah.
like a phenotype to a clinical.
Yeah.
Would they be able to look at blebs with current technology?
Well, so it was interesting. So when the paper came out, I got in one day 10 phone calls by hemonc colleagues throughout the US, right? And all of them said, if I see leukemia cells blebbing in the bone marrow, I know I cannot do anything anymore for these patients, right? And so I still have sort of a rogue academic shop in Dallas, and I have a postdoc there looking now into this, and it is amazing how that is. So the blebbing has been ad hoc observed and occasionally described, right? But now I think we have a mechanism really worth pursuing. Yes, please.
So, what keeps these cells?
I said I don't know. So that's again, experimentalists. That's potentially a heyday. Yeah, I don't know. Okay, good. Thank you very much.